Number 10, bacteriophages, temperate phages. Slide 19 of our second set of virus slides. So we are going to look at bacteria phage lambda as a model for the temperate phage. So let's look at our video showing the temperate phages infection cycle.
When phage lambda infects E. coli, either the lytic or the lysogenic cycle may be followed. In both cases, the first step involves the phage attaching to the host cell and injecting its DNA into the host cell. In the lytic cycle, phage nucleic acid is replicated and phage genes are expressed, resulting in production of phage proteins. Mature phage particles assemble and the host cell lyses, releasing the phage particles. In the lysogenic cycle, the phage DNA is not replicated or transcribed. Instead, the phage DNA integrates into the host cell genome. The host cell can then replicate, carrying the integrated phage genome. The integrated DNA is referred to as prophage DNA, and the host cells carrying the prophage DNA are said to be in the lysogenic state. When the cells are exposed to ultraviolet light or to certain chemicals, phage induction occurs. The prophage DNA is excised and the phage enters the lytic cycle.
Okay. Temperate phages, or also known as lytic. So when the virus first encounters the bacteria, it'll attach and it'll inject its genome, and then the virus has kind of two options. And what triggers these options is probably a lot of complex environmental factors. First off, the virus could act just like a lytic phage, orchestrate production of virus pieces, put those pieces together, cause the cell to lyse and release. So in this mechanism here with the blue line, very, or indeed near identical to the T4 lytic cycle. But what they can do alternatively is that they can integrate their DNA. So in this way, they are quite similar to those retroviruses we looked at in animals that can integrate their DNA into our DNA. If the virus chooses this route, integrates its DNA, then we call it a lysogenic infection. So this bacterial cell has a lysogenic infection, and as a result, we can call this bacterial cell a lysogen. It kind of sounds kind of a little bit tricky to remember that the cell itself is called a lysogen. The DNA that's been integrated into the host genome then often just rides around inside the cell. It rides around as the bacteria goes through cell division, as the bacteria goes through binary fission. It will duplicate the viral material along with its own chromosome. Just like in a human cell, the human cell has no mechanism to identify what is human genome and what is provirus. The bacterial cell has no mechanism to distinguish between bacterial genome, and in this case, prophage. So our bacterial genome is a circle. the virus comes in, snips the bacteria's genome and inserts its own piece of DNA. When this happened in our cells, we called it a provirus. In this case, it's called a prophage. The virus prophage has on it genes that were brought in by the virus itself. Take a look at the implications of a virus inserting not only its genetic material, but inserting new genes into the bacteria's genome. So on to slide 22. Here is that cycle once again. Just a quick note about the structure of our lambda phage. Inside the virus particle, we have a double-stranded linear chromosome. Once it's injected into the bacteria, it forms up into a circle, and then that circular DNA sticks to the host cell chromosome, and they combine into a single piece of DNA, i.e. the chromosome. And then the lysogen, the bacteria that now has the prophage in it, will undergo binary fission, and it'll replicate the viral genome along with its regular genome. So this is just a very kind of simple mechanism for a virus to just ride around throughout time. It gets into the bacteria, and then all the virus does is just exist as a piece of the bacteria's genome. And the bacteria is responsible for replicating and duplicating and packaging up. So requires absolutely no effort whatsoever from the virus. So we have a virus kind of like a stowaway, like a stowaway on a boat. Our stowaway is just riding around inside the boat, not contributing to any activity, just lying there. But if the boat starts to sink, then our stowaway wants to get off the boat and find a new vehicle, a new boat. So our lysogen, our cell with the prophage, if the cell gets damaged in particular, the prophage can snip itself out and you can start into the lytic infection, where the purpose is to make lots and lots of copies and escape the cell. The DNA of our temperate phage is inserted into the DNA of its host using an enzyme called integrase, and integrase is encoded on the phage genome, so it does require a little bit of decoding first before the viral genome can be inserted. Entry into the DNA is quite specific, and in general bacteriophages are themselves very specific, whether they be lytic phases or temperate phases. Not phases, lytic phages or temperate phages. There are lots and lots of different kinds of bacteria, and there are lots and lots of different kinds of phages. So E. coli, for example, will have its own set of phages. And an E. coli phage will only ever stick and inject into an E. coli. And a different kind of bacteria, maybe S. epidermis, that will have its specific phages that will only infect S. epidermis. So an E. coli phage will not infect anything else, and S. epidermis phage will not infect anything else. Once integrated, the viral genome in the DNA host genome is called a prophage. It replicates with the host. It can be excised. Excise just means cut out. It can be cut out given certain triggers, and that cut out phage genome can then send the cell down the lytic pathway. One important aspect are those genes that the viral genome brings with it. Sometimes infection of a bacteriophage in a bacteria isn't necessarily bad for the bacteria. In fact, the virus, in inserting its genome and giving the bacteria a new set of genes, those genes then are transcribed and translated by the bacteria as it goes about its kind of day-to-day business. They can be transcribed and translated into proteins that alter the bacteria's ability. When a bacteria is altered by the genes that are brought into it by a bacteriophage, that's known as lysogenic conversion. There's some examples of some organisms that get converted on page 341 in the table there at the top. So one example is E. coli. E. coli in your digestive tract, doing no harm, in fact probably doing you good, but we've all heard of the nasty kind of E. coli. That's probably going to be E. coli 0157H7, the kind that makes people very sick. The reason why this particular strain of E. coli makes you sick is because it has the ability to produce a toxin called a sugar toxin. And the gene that makes the toxin isn't kind of native to the E. coli. That gene is put into the E. coli by a virus. So really, when you get sick from E. coli, you shouldn't blame the bacteria. You should blame the bacteriophage that gave the bacteria the ability to use the toxin. Another good example is diphtheria. Diphtheria disease is a pretty nasty disease, but the bacteria that causes diphtheria is only able to have this nasty effect because of the toxin that it is given by the bacteriophage. And more specifically, the bacteria is given the gene as the bacteriophage incorporates its DNA as a prophage. And now the bacteria has this new set of genes. One of those genes is transcribed and translated into the toxin, and it's the toxin that goes on to harm the person. So I guess the idea is that if the virus can make the bacteria more successful, that means that there'll be more bacteria, and the virus prophage will be passed on to all those multiplying bacteria. So it's a win also for the virus. So some of the mechanics of the phage, the lysogenic infection, involves the ability of the prophage to get in and then also to get out again. The ability of the prophage to leave the host and begin the lytic cycle is based upon various enzymes that the phage itself encodes. The phage encodes for an excision enzyme, an enzyme that's going to cut the phage out. So in order to get out of our host genome, the phage needs to be cut at either end so it can escape. And the enzymes that cut out the phage are made by the phage itself. But in addition to the snipping enzymes, the phage also makes a repressor that prevents the snipping enzyme from doing its job. So whilst the repressor is there, the phage remains inside the host because it can't be snipped out. Something happens then to the host, particularly things like DNA damage from UV exposure. That DNA damage switches on host systems to repair DNA. One of the things the host systems do, called the SOS repair system, is it destroys the repressor protein. It snips up the repressor protein. And now our viral snipping protein is active. It'll come along, it'll snip out the prophage, and it'll be able to go along and start the lytic phase. So this is how the lambda phage or the prophage knows when the ship is sinking. The bacteria gets damaged, that leads to a chain of events, and that leads to the virus escaping the sinking ship. Now, whilst a bacteria has a prophage, it's in the best interest of the prophage to maintain the bacteria, maintain it in a healthy state. Healthy bacteria keeps the prophage inside it, keeps on replicating, passing on the prophage just through time. So one thing that is an interesting feature of a prophage infection is that it then becomes much harder for the bacteria to be infected by another virus. It's in the prophage's interest to keep the bacteria safe, so if a virus comes in, if a lytic phage comes in that's going to destroy the bacteria, the prophage wants to stop it. It'll do that by preventing the incoming phage DNA from expressing any genes. The other thing it might do is that genes in the prophage may alter receptors. So the prophage genes contain altered variations of the stuff that's in the bacterial membrane. And we've seen before that the process of virus sticking into a receptor is very shape specific. They either do fit or they don't fit. If the prophage changes the shape of the receptors, it means that this incoming virus can't stick. If it can't stick, it can't make or it can't affect the cell. If it can't stick, it can't inject its genome. Okay, so infection mechanism of a temperate phage. What is a prophage? What is a lysogenic state? What is a lysogen? Know the decision the phage has when it first gets in the cell. Does it go to the lytic cycle or does it go to the lysogenic cycle? Know what triggers the excision, the snipping out of the phage DNA. I guess know what's in it. What's in it for the phage just sitting there in the microbes, the bacteria's DNA. Know what the term lysogenic conversion means. This is when the bacteria is altered by the genes that were inserted from the phage. Can be good for the bacteria, can be very bad for the host. Superinfection, i.e. the infection of the subsequent infection of the cell by another phage. Know how a lysogen prevents this.